Hi students, how are you? I hope each one among you are doing great and well. Welcome to Supnet's classes. This is Rahmat Latif, your biology educator. In this video, we are going to learn about digestion and absorption. In our previous video, we just saw what are the different digestive glands inside our body, like we have salivary glands. I'll write it here. We have salivary glands, we have liver, we have pancreas, which acts as different glands inside our body, which their ultimate goal is to digest the food products. They help in digestion. Okay, I hope this concept is clear. Then we studied about stomach, that in stomach we have HCl, hydrochloric acid, whose what we call it as whose pH value is 1.8, right? pH value of HCl is 1.8. We learnt about renin. Renin is proteolytic enzyme found in milk, found in stomach of infants, right, which helps in digestion of proteins, helps in digestion of proteins in infants, right? I hope this concept is clear. Okay, moving forward, what we are going to learn about digestion in this session, let's see. Okay, I told you about peristaltic movement as well. I told you that through mouth, in mouth food gets converted into bolus. In mouth, food gets converted into bolus, which is then transported into the thorax region, which ultimately leads it to esophagus, and through esophagus, food gets into stomach, through which movement? Through peristaltic movement, right? Peristaltic movement. This movement is wavy movement. Okay, this is peristaltic movement. I hope this concept is clear. You know, inside our body, inside us, various types of movements are generated. First movement is this peristaltic movement which took place in esophagus. Similarly, we have muscular layer, right? We have muscular layer inside the small intestine, which helps in generation of various types of movements. And what happens through those movements, what will happen, that will ultimately help us to mix the food thoroughly, right? These movements of muscularis layer of small intestine will help in mixing up of the food, okay, and thus digestion will take place simultaneously. As I already mentioned that we have bile juice and pancreatic juice. They are transported through a duct known as hepatopancreatic duct, right? And with the help of this duct, this food is transported to small intestine. These nutrients, this is transported to small intestine, and this hepatopancreatic duct and small intestine is guarded by a sphincter known as sphincter of Oddi. Sphincter of Oddi. Okay, I hope this concept is clear, right? Okay, moving forward, we know that, as I already mentioned, that pancreas is mixocrine gland. It is both endocrine and exocrine. So some enzymes are secreted by in pancreatic juice. We have some enzymes. Some enzymes are present in our pancreatic juice. Okay, let me erase it. Enzymes are present in, we have some enzymes present in pancreatic juice, like we have trypsinogen, chymotrypsinogen, we have trypsinogen, we have chymotrypsinogen, right? We have trypsinogen, chymotrypsinogen, procarboxypeptidase, peptidase, we have lipases, we have nucleases, so on and so forth, right? And please remember that trypsinogen is activated by an enzyme. Trypsinogen is activated by an enzyme and it gets converted into trypsin through which enzyme? Through enterokinase. Through enterokinase, trypsinogen gets converted into trypsin. Okay, I hope this concept is clear, right? And other enzymes are also present in pancreatic juice, as I already mentioned. I'll highlight it here as well. We have trypsinogen, we have chymotrypsinogen, we have procarboxypeptidase, amylases, lipases, nucleases, and I already mentioned that trypsinogen is activated by an enzyme known as enterokinase into active trypsin, right? And trypsin in turn activates other enzymes which are present in pancreatic juice. Bile is secreted now by hepatic cells which are actually present in hepatic lobules of liver, and that bile contains bile pigments. Please remember this. This reaction is very important as it comes in PYQs of Neet. Okay, Neet examination. I'll write it again. Let me write it again. Trypsinogen gets converted into trypsin with the help of enzyme known as enterokinase, and this trypsin activates other enzymes of pancreatic juice. Now talking about bile contains bile pigments. Bile contains bile pigments like bilirubin, biliverdin, cholesterol pigments. Okay, it contains so many pigments. It contains cholesterol, lipids, but it doesn't contain any enzyme. Please remember, pancreatic juice contains enzymes, so many enzymes, which is highlighted here by pink highlighter, but bile does not contain any enzyme. What's the function of bile? Bile helps in emulsification, breakdown of fats. Okay, please remember, bile helps in emulsification of fats. Okay, bile helps in emulsification of fats, breakdown of fats, and into very small particles, as we know. And moreover, bile also activate other lipases which helps in emulsification of lipids. Okay, I hope this concept is clear. Okay, moving forward. As I already mentioned in our previous session, that we have goblet cells in our intestine which secrete mucus, and secretion of these goblet cells constitute intestinal juice or succus entericus. Inside our intestine, we have goblet cells which secretes mucus. It secretes mucus, okay? And this mucus, we have so many, secretions of this intestine, which ultimately forms intestinal juice. Please remember the another name of intestinal juice, that is succus entericus. Succus entericus. Okay, I hope this concept is clear. This juice, this succus entericus, this intestinal juice contains lot of enzymes like disaccharides, maltase, dipeptidases, lipases, nucleosidases. Okay, maltase will break down maltose, dipeptidase will break down peptides. Okay, lipases will break down lipids, fats, nucleosidases will break down nucleases like RNA, DNA. Okay, please remember that we have intestinal um, pH of intestine is 7.8. Okay, okay, moving forward. Okay, moving forward. Proteins, we have so many like proteins, proteoses, peptones. They are partially hydrolyzed proteins. Okay, in the chyme is the juice of stomach. Okay, juice in the sense the digested particles there is known is that semi-solid semi-liquid substance like substance, that's why we call it as chyme. And proteins, peptones, peptides, proteoses, they are in pancreatic juice, they are acted by so many, what we call it as so many enzymes, and thus they are divided into smallest particles. Okay, I hope this concept is clear, right? Very nice. Okay, you have to refer some reactions on your NCRT, on your textbook. Please remember that you have to refer some reactions on your textbook, right? Like what is the end product of proteins, peptones, proteoses. We have dipeptides, right? Then similarly we have carbohydrates. Carbohydrates are hydrolyzed by pancreatic amylase. Carbohydrates get converted into disaccharides. Carbohydrates gets converted into disaccharides. Then these disaccharides gets converted into monosaccharides. Carbohydrates get converted into disaccharides. Disaccharides get converted into monosaccharides. Similarly, we can say polysaccharides. Carbohydrates, another name of carbohydrates is polysaccharides, right? Or we can say starch. They are acted by amylase. They are acted by amylase. Similarly, we have fats. Fats, lipases acts on fats, and fats get converted into diglycerides. Diglycerides then gets converted into monoglycerides. These are the simplest substance which are formed, right? Great. Then we have nucleases. In nucleases we have what we call it as RNA, DNA. Okay, nucleases, sorry, nucleic acids. Nucleases are enzymes. Nucleic acids, be that ribonucleic acid or deoxyribonucleic acid. Nucleic acid or nucleic acid, nucleases acts, thus they get converted into nucleotides. These nucleotides further get broke down into nucleosides. Okay, I hope this concept is clear. Please, you have to remember these reactions, not remember. If you know which are the smallest products, like in we have, you might have studied in, okay, I didn't teach that, taught that chapter yet, biomolecules. You will learn in that which are simple products, which are complex ones, okay. But for now, please remember them, okay. I hope this concept is clear, right? Okay, I think it's enough for today's session. Please, again, I'm saying, please revise these reactions again so that they will benefit you. Okay, I hope this concept is clear. I'll meet you in any other interesting session. Till then, goodbye, take care, thank you so much.